So Synpromics, so we are a gene control company. What we're focused on is, in gene therapy, what we're trying to do is deliver genes to treat diseases. And the question that we always pose is, well, once you've overcome the challenge of delivering the gene, then you're then faced with the challenge of how do you control the gene once delivered? And just to sort of think about this a little bit, because the world of gene therapy has spent the last 20 years or so trying to resolve these challenges of how you deliver the genes in vivo and into particular cell populations. And so there's still a lot in people's minds about focusing on the capsid and what the capsid can achieve. And I think if you're trying to think of missions to Mars, the first challenge you've got to face is, well, how do you actually land on the planet? And that's the challenge that gene therapy has been focused on very much. But what I'd like you to think about is, once you've actually landed on Mars, what are you going to do when you get there? And how are you going to design a rover that's going to be able to go out and really investigate and interrogate and generate knowledge? And so when you think of the expression cassette and how you control the gene and get optimal functionality of the gene once you've landed in the cell, that's the issue that we're trying to tackle and provide solutions to. So to make sure that once you've landed on Mars, you actually getting value and interesting data. And so what we're focused on is the expression cassette. A key component of the expression cassette is the promoter. And we're developing and designing unique, sequence-unique and functionally unique synthetic promoters that ascribe specific attributes to the control of that gene for safety and therapeutic purposes. It's not just about the promoter. There's other components of the expression cassette, like the UTRs, like the introns, etc. So we're also working on optimizing those. So in that context then, why focus on the promoter? What's the expression cassette going to give in terms of value? And I'd like to sort of describe that by using this slide here. So on the left-hand side is where I would say gene therapy is today. It's really focused on monogenetic diseases and constitutive expression of those diseases, often using endogenous or viral promoters. With a synthetic promoter, what we can focus on doing is creating expression cassettes and creating promoters that are highly selective to the target tissue, much more selective than the capsid will be. While the capsid might get you into a particular organ, if within that organ you need to get into a particular population of cells within that, you can achieve that expression through the promoter. It's also about being able to design the expression cassette to get expression that's optimal to the disease. So you're not dictated to by the promoter that you've been using, but you can custom build the promoter to the expression strength that's required. And it's also doing other interesting things like addressing size constraints and addressing multi-gene control. So that's sort of generation one of where the industry has been focused. And what we're starting to think about is, where does gene therapy go next? How do you start to get out of the small monogenetic rare disease indications and start to think about larger market opportunities? And here there are challenges that the industry will face, both over safety as well as efficacy. So we've been developing and designing regulatable gene cassettes. So you can use small molecules or environmental conditions to turn genes on and off. So you can turn the gene, you can induce the gene, but you can also repress the gene. And then we're also then starting to think about how we enable more complex diseases that may require multi-gene approaches. So how do you end up with an expression cassette with multiple genes with the appropriate stoichiometry of expression from a selection of promoters? And then we're also creating biologically responsive promoters. There's promoters that will respond to a disease state, for example, as a future generation of gene therapy for more complex diseases. So that's our vision of where we see the initial technology ultimately leading towards. As a company, we're also evolving. So we've very much started out as an early stage company developing the platform. We're a bunch of great bioinformaticians, molecular biologists, machine learning scientists, really using AI and our knowledge of both the machine learning skills and the molecular biology skills to develop the technology. We've taken this platform now and validated what we're able to do both in an in vitro setting and now in vivo setting, and uniQure now adopting our promoter in their Heme program. So we're now ideally positioned as a company to move from investment in the platform to start to develop our own pipeline of therapeutic products. So in that regard, we're going through that transition. We're out now actively speaking to investors and raising finance to enable us to do that. So if that's the story of what we do, then I want to quickly convince you that we can do it by showing you some data, because we're scientists at the end of the day. Data is what speaks. So here's just one data slide that uniQure have presented. So what uniQure for haemophilia A, what they were looking for was a promoter that was specific to liver in terms of its activity, that was small in size because there's a packaging constraint with Heme A. So we created a promoter that was only active in the liver, that was in the region of 200 to 250 base pairs in size. So in promoter terms, this was incredibly small. And what they were able to show in a non-human primate study is that our promoter gave them an 8-fold increase in productivity from that gene expression cassette than their internal comparator. So a huge bonus in terms of yield as well. And that's now heading towards the clinic. On the inducible side of things, we've created this liver-specific construct that we can regulate using a small molecule. And so we've been able to show that this works in vitro, and now we've got in vivo data in the mouse showing that we can induce expression of a gene with either a 9-hour or a 24-hour cycle in a dose-responsive manner. We've also then been able to do the reverse, which is to create a liver-specific repressible promoter control element. So the gene in its natural state is being expressed, and by dosing with a small molecule, we can turn it off temporarily. As well as the liver, we've been doing a lot of work in the CNS, and we've been getting some really exciting data over the last few months to show that we've been able to create a pan-CNS promoter that has much broader spectrum of activity to a higher level of activity than the standard which is currently used, which is the synapsin promoter. But here, what you'll see in some of our other iterations is that we're being able to create promoters that represent a range of different strengths of expression across the CNS. Our ultimate goal, though, is to be able to target specific subpopulations within the CNS. And this is an example of where we've been targeting specifically expression only in dopaminergic neurons. And through our latest set of in vivo data, we've been able to show that you see the dopaminergic neurons highlighted with the red stain here, and the activity of our promoter highlighted by the GFP strain, showing that we've been able to specifically target with a promoter gene expression in a dopaminergic neuron. And that's a project that we're doing with UCL, targeting a particular form of Parkinson's, infantile Parkinson's disease. And then finally, we're also consciously aware that bioprocessing bottlenecks are huge in gene therapy. So we're using our inducible platform to create stable producer cell lines for AAV, where we're basically taking the triple transfection constructs, putting those genes under inducible control and stably transfecting them into a 293 cell line, and using induction as a way of being able to turn on AAV production from that cell line. And we anticipate, I've not shown much in the way of data here, but we certainly anticipate at ASGCT being able to talk about that in more detail. So really the summary of the company is we're focused on how do you get maximum value from the payload that you deliver. maximum value, but also safety and maximum efficacy, and by focusing on the expression cassette and gene control to be able to do that. So we really stand on the shoulders of delivery. We stand on the shoulders of the capsid technology. To date, we developed the business through a partnering model, but now we're transitioning to start to develop our own therapeutic programs and raising the capital to allow us to do that.
Thank you, David. I like the analogy of landing on Mars. I also heard another analogy about traditional vector gene therapy being sort of like a Lamborghini with a Model T engine, where the promoter is sort of the engine. Could you talk a little bit about DNA and how much codes for proteins versus elements for gene expression, or why it makes sense to focus on promoters as well?
Yeah, so I don't know, it's known to me, but not always generally acknowledged, that actually the coding DNA, only about 2% of the genome is actually coding for protein. And there's about 98% of the genome that people originally thought was just sort of dark matter. It's there as scaffold, it was just supporting. But as we interrogate increasingly, we realize that actually there's a complex network of control, which immediately makes sense, because if you imagine every cell carries the same DNA source code, but obviously every single individual cell, the reading of that source code is entirely different. And not only that, but you've got genes that are being orchestrated to come on at particular points in time, to be silenced at points in time, to be regulated, to be responsive to the environment. So there's a huge complexity in the way that genes are controlled. And so our bioinformatic approach is to basically gain all the learnings we can from what the cell does naturally and pull out those parts of the DNA that enable that level of control, and then start to recombine them and re-engineer them in interesting and novel ways to create something that essentially we could be given a target product profile or a design brief for a promoter or for a gene expression cassette, and then we'll use the bioinformatics to design sequences that enable that design brief to be achievable.
Right. So your focus on big data has enabled solutions that it seems like I think you have something like partners in the teens or quite a few formidable gene therapy companies. But why the shift in focus now to go internal? Why do you think that's a good strategy for Synpromics?
It's a good question, and sometimes I talk to people and say, yeah, well, that's obvious. It's where are we going to create the value, and so. We have successfully built the business through a partnership, generated data, and those partnerships are hugely valuable to us and will continue to be. But we're starting to see some of the unique features of our technology that we think, actually, this is so exciting that we want to keep some of that value for ourselves, but also be aware that to create maximal value, there will continuously be an iteration. It's not that we could just build something and hand it over to somebody else, but there will be learnings, there'll be feedback, and there'll be an iterative process to get to the optimal therapeutic. And we believe that's best done in our hands than simply handing something over to somebody else.
Great. I'm a fan of science, and I recall your company has quite a few Ph.D s. So what's that number, first of all? And second of all, what do you think is the source of hidden value that maybe investors should appreciate about your story? Or what are they missing?
Yeah, so I am unashamedly say that we are a bunch of real nerdy scientists. And when you take a multidisciplinary team of bioinformaticians, physicists who are doing a lot of the data mining, the people who are doing the machine learning, combine them then with some really great postdoc molecular biologists, they then pass on their know-how to the cell biologists, who then pass it on to the vectorologists and the virologists. So we have all of those functions within the company, and almost everybody has got several postdocs under their belt. So we're a huge engine of scientific innovation. So that's great. It's a delight to work with the team. I think in terms of if people are missing anything about the story, I think it's starting to really think, well, gene therapy we're getting some great data in monogenetic diseases today, but where's it going to go? And how do we start to have a vision for the future, for larger market sizes, for indications that's going to start to get large pharma even more excited. And to enable that future, you need to have the right basic understanding of science and have the right tools built. And we're focusing on making sure we've built those right tools to enable the next generation of gene therapy products.
Great, thanks, David. And I suspect Synpromics is a name that many people will hear a lot more about in the future.
Thank you.
Thank you.
Thanks.